Good morning. Welcome to the PrevMed Dr. Ford Brewer Show on the YouTube channel live. Thank you all guys for joining us this morning. As you can see, I am not Dr. Ford Brewer. I'm James West. I'm sitting in for Dr. Brewer this morning, who is traveling, flying to warmer climes on the Gulf Coast, and was actually in an airplane as we speak. And so we're covering the show today, but we have a great show for you. I think you all will enjoy it. You have in the past had Dr. Vigorist on the show. We've interviewed him a couple of times. He always does a great job. Dave is an old and good friend and one of the smartest individuals I know. Of course, I tend not to hang with a lot of smart people, Dave, so that may be a very low bar. Dr. Brewer is give him a quick introduction, and then I'll run through some points about PrevMed. Dr. Vigorist is on the medical school at Vanderbilt University, as well as being the chief science officer for a number of biotech companies around the country. He helps develop assays, as well as develop studies and courses and biomarkers. And so he brings a broad wealth of information about the biotech space, as well as what we do in prevention. So Ford and Dave and I have worked previously on two different ventures, one being Epigenetics, or Lifetime Sciences as it's called now, which was a biotech company, early stage human genomics firm out of Nashville, Tennessee. And he continues to work with a number of these COVID-based labs and developing immunoassays. So we'll have him in just a minute speaking to us. Our topic today later is going to be genetics and the disease of type 2 diabetes. So I think you'll find that very interesting. I'm going to pull up the PowerPoint and start walking through this morning, because, as we said, our topics are going to be specifically TCF7, L2, ATM, and Haptoglobin, which are three very important markers in treating and recognizing the broad spectrum of diabetes risk and treatment. So if you're listening, if you know anyone with diabetes and are interested in diabetic topics and genetics, you might grab them, tell them to jump on the channel and watch this as we get going. As you know, PrevMed continues to be a source of teaching people like yourselves about the risk of inflammation, the risk of diabetes, heart attacks, strokes, dementia. And you can go to our website and you're able to download any number of the webinars that we currently have, courses that we make available to you, or just sign up and consider being a patient. We do see patients on a private basis. You can see Dr. Brewer as your own physician. And so if you register now, you can get a discount from that. So we look forward to you doing that, or signing up for the courses such as the insulin resistant course, heart attack and stroke prevention, cardiovascular information. We're going to talk a lot about cardiovascular information this morning, and even the plaque evaluation, the ability to reverse arterial plaque, very important topic. Again, you may ask Dave some about that. We did a lot of research on that. And as we said, you can have a membership subscription to sign up to see Dr. Brewer as your provider. Dr. Brewer is making a lot of progress. There will be a book coming out soon, What Stress Can and Can't Tell You. Look forward to seeing that. And then, of course, we're refreshing our website and looking at that. Now, understanding that, let's talk a little bit about diabetics and genetic screenings, and particularly the TCF7-L2, ATM, and Haptoglobin with Dr. Vigorist. So we'll start that. I'll go ahead and kick it back to Gilbert. Gilbert is going to do our intro, and then we'll get started with Dr. Vigorist. All right, good morning, Dr. Vigorist. I assume you can see and hear us well. You've joined us, I assume, from Nashville, Tennessee. I recognize the room you're in, so welcome to the show again.
Thank you very much. Appreciate being here.
All right, as many can see our screens, as we said, you're well educated. joke with both you and Dr. Brewer, both of you, between all of your fellowships and your postgraduate studies, the two of you spent a whole lot of your life in a classroom. And so I just summarized a couple of your degrees. I didn't go into all of your background, but always being on the show, I also wanted to remind everyone that I did graduate Miss Walker 6th grade manners school. So you're not the only educated person on this show today. So
you continue to wisely use everything she taught you, right?
I try my best. I try my best. All right, let's talk a little bit about genetics. As you can see from the screen, I want to give a baseline first. Many of our listeners, we have some great listeners here. Many of them are providers themselves, scientists, as well as patients. But at the risk of maybe we do or maybe we don't all have the same basic understanding of the foundation of genetics and diabetics, let's talk a little bit about DNA. We use that term a lot. And this is not an exhaustive, this is a very basic illustration that I use for myself to understand, to lay the groundwork for what we're going to talk about with these three particular genetic variations that we're going to discuss. But DNA, of course, is just the building block of life. We can oversimplify it and just say it's the code. It's the computer code for our cells, and contained within the nuclei of our cells are these chromosomes. And on these chromosomes, we have our genetic makeup. And we've seen the double helix. It was discovered in, I guess, 100 years ago, but really started coming to the forefront in the 1940s and 50s with Glick and Watson and Franklin and others, and discovering the composition of these. But for me to understand it, if you see the screen, I describe DNA as these base pairs, these crossbars that connect two intertwined ribbons. Those ribbons, of course, made up of phosphate and deoxyribose. And then between those ribbons, you have your base pairs. Those base pairs are made up of 4 different chemicals, as we see. We call it A, T, G, and C, or A, T, C, and G. Each of those have their own unique chemical marker, and they pair up in base pairs in order to start creating the base of these codes. And when you put 3 of those together, 3 of those chemical compounds, each 3 creates, if you would, a unique word. And those words are the word instructions to the cells to tell proteins what to do, to replicate or direct the function of the cells. And then when we talk about genes, genes are just sections of that DNA. And I try to, in understanding myself and giving a baseline explanation, maybe slightly oversimplified so our audience that are sophisticated can overlook that. But I always say a mutation, trying to understand the difference between mutations, deletions, duplications. A mutation is if A, T, G, C, 3 of those combined make up a word. Then a mutation is when someone in your instructions has misspelled a word. So you may have a mutation where the A should be a C or the G should be a T, and that mutation has changed. Sometimes changing the word rot from rat, R-A-T, to R-O-T, changes the meaning completely of a word. While at other times, someone can look at the change and still get the meaning of the word. Well, mutations in our genes are very similar. Sometimes it's significant, the change that occurs, and sometimes it's not significant. Rarely is a mutation good, if ever, but it does occur. First, it's deletions in the strand of genes and the DNA. A deletion is like someone was copying a book or writing a book, a manuscript, and they just forgot a word. And again, forgetting that one word can be meaningful, or forgetting that one word may not change the meaning of the sentence at all. But a deletion is you've missed part of that code. And a duplication is like when you're copying something on the copy machine and you get an extra page, the same page copied over again. So occasionally you buy a book, and when people bought books and still flip pages, I know everything's electronic now, but I still like picking the book up and reading it. And every once in a while there's a mistake in a book where they made the copy of the same page. And you look, you flip, you go, well, they just made a mistake. And on so many books, they copied it. And then if someone ever copied that book again, they'd probably make the same mistake, make that copy over again. And so duplications are very much like that. So what we're going to talk about today are some mutations, some deletions, some duplications within the genome makeup of an individual, particularly someone with diabetes. So Dave, do you want to comment on that, clarify that, clean my mess up? I have any comments whatsoever?
Sounds fine.
Dr. Brewer, the professor, and Dr. Vigors, professor, has taught me well. All right, Dave, I'm going to hand it back to you. I've got control, but if you would be so kind to walk people through now the genes and the discussion of these three particular candidates that we're discussing relative to diabetes.
Sure. So what we're going to talk about today are three genes that have a relevance at different stages in the development of diabetes, the management of diabetes, or the consequences that may happen. So, whether you have additional issues like cardiovascular disease or stroke risk, those sorts of things that are exacerbations as a result of diabetes. So TCF7 is a gene that was discovered a number of years ago, and it was discovered in several very large surveys in the United Kingdom and in Scandinavia. And what TCF7 is it's a transcription factor. So when you look at how do you read and how do you interpret the DNA, we have a number of additional proteins that help to make that read clear and understandable. And one of the things that gets done is transcription factors are responsible for turning on and turning off a given gene. So, you get a signal from the body, you get a signal through the cell, and it turns on the transcription factor, and it tells your cell to make a certain protein, or it tells your cell to stop making a certain protein. So the discovery around TCF7L2 is that it turns out right now to be perhaps the susceptibility gene with the largest impact on the development of diabetes. And the reason for that is that TCF7 regulates the expression of insulin. So this is a transcription factor that does most of its work within the pancreas, and it turns on or turns off the expression of insulin. So if we were to take this transcription factor and increase its function, so basically you get a gain of function. So we turn it on and we over rev it. What you end up seeing is you get a stimulation in the secretion of insulin. And conversely, if we were to shut it down, if we were to silence it, you would get a reduction or a suppression in the amount of insulin that's produced. So if you have a variation in this particular gene, you may be incapable of properly regulating the expression of insulin. So when you need the insulin, your body can't turn on enough of it to compensate and to do its biological function. And by the same token, if you've got too much of it, then your overproduction of insulin is going to cause you problems physiologically as well. But this is just one factor in the development and the management of insulin. As you've probably heard from Dr. Brewer, there are a multiplicity of things that can go wrong and can lead to the development of insulin. It could be something like this, where there's a mistake or mutation or a dysregulation in this transcription factor. It could be antibodies that you produce against the cells that produce the insulin. So if for type 1 diabetes, for example, there is some evidence out there that we produce autoantibodies against the cells that produce insulin. So there's a number of factors that go into developing diabetes, and this so far is turning out to be one of the strongest ones. The unfortunate thing is that very few labs, if any, are running this now on patients for clinical interpretation. So, if you had a history of diabetes in the family, or if you just wanted to know, do I have an increased risk? There are very few places to get this test, but the test does have a tremendous impact on determining whether you have a higher risk of developing diabetes. So it's one of those check marks that says, hey, this is wrong, this is wrong, this is wrong. And that could be diet, it could be autoantibody production, it could be transcription factors, it could be a mutation in your insulin gene where it just doesn't produce like it should.
Right. Now, pardon me of interrupting, but it's a good time to point out the term epigenetics. We use that, and many people may have heard that. but that's a good illustration. You discussed turning the switch on, turning the switch off. So just because you may have a transcription factor 7 doesn't necessarily mean, it doesn't guarantee you may or may not develop diabetes. You may overproduce or underproduce insulin, but it's that epigenetic event. And by that we mean what? That you do things that make it more likely to turn on.
There are things that happen to us over the course of our lifetime. So we start off with a genetic profile, and sometimes that genetic profile has a mistake in it, a duplication, a deletion, a mutation. But then on top of that, we have an additional set of regulation that happens that comes as a result of environment, for example. So epigenetics could be things that happen because of diet, because of environment, because of chemical exposure. And most of the time, epigenetics is the addition of a methyl group. So that methyl group gets on top of the gene and can artificially turn it off. And it doesn't allow the transcription factor to come in and do its job. So you can kind of think of us as one really large system of switches, on and off switches. And both of the two proteins, or the two genes that we're talking about here to begin with, TCF7 and ATM, are both switches of sorts. So they help to turn on or turn off, and they have to be regulated by other outside influences. They're regulated by glucose. They're regulated by epigenetics. They're regulated by environment. So, having all of these switches going on and off continuously to turn on and turn off things means that we have an exquisitely complex physiology that most of the time works really, really well. It doesn't have too many glitches in it, but occasionally a glitch develops, either as an inherent mutation or alteration, or as a result of epigenetics, because we've been exposed to something that turns the switch off or turns it on.
Like pancakes.
Yeah. So
go ahead, keep going. I switch slides on you there.
So TCF7 is a very strong genetic component. I don't think there's anything out there that seems to refute that it has a role. It's got a very powerful role. It's been looked at in a number of different populations around the world, and all of that research has held very strong that there are areas within this particular transcription factor that have a dramatic effect on the pancreatic beta cells, and that impacts insulin secretion.
Right.
So you turn off that transcription factor and you lose the ability to properly secrete insulin.
And that's what we're doing when the viewers hear us talk about a Kraft insulin score. You're really trying to see that, whether it's because of a predisposition with TCF7L2 or something else. We're trying to get a view of that in real time. And the Kraft insulin score, for those of you who have done it, OGTT is another term for it, but the Kraft is more of the view of what is happening in the OGTT, the insulin, the glucose study. So we pound a person with lots of glucose by having them drink that nasty glucola drink, or anything with high amounts of fructose or glucose. And then we have a fasting score, so we get a look at what is your glucose level, what is your insulin level at baseline fasting, and then you look 1 hour, 2 hour, and sometimes 3 hours. And so, for instance, you're saying if you have this transcription factor 7, it's very likely that, depending on whether it's revved up or revved down, you're either going to oversecrete insulin, so you get an overreaction, which has one physiological impact, or in most of the time, what we're seeing with people with diabetes is they're undersecreting that insulin, and so their blood sugar levels continue to remain high, which continues to impact the inflammation cascades that occur in their bloodstream.
Right. We end up having significant damage as a result of that high sugar.
And we're trying to, with Kraft, we're scatter charting and looking at 0, 1, 2, 3 hours, and looking at 2 or 3 different parts of the seesaw to oversimplify. If it takes an enormous amount of insulin, a large reading of insulin in order to affect that glucose. That's telling you one thing. If it's also telling you if your insulin response is normal and your glucose level is normal, then you're not insulin resistant. But if you cannot produce enough insulin, or you overproduce insulin but still doesn't change your glucose rating appropriately, then we get a good feel of your insulin resistance or your full-blown type 2 diabetes.
Right. There's this balance we have to try to keep between the glucose level and the insulin, right?
Right.
So there's this continuous biofeedback. And these two first genes are participants in that biofeedback. So this is the first one. Right. So if you were essentially a normal, healthy person, you don't have any real issues back and forth with, too much glucose or too much insulin or vice versa. You could use TCF7 to determine, do you have a risk? And that means you could do something proactively, right? You could say, all right, I know I had a risk. It's one of maybe several, but these are the things that I can do to proactively alter the ability of, not the ability, but alter the possibility of becoming insulin resistant or becoming diabetic,
right.
One with a diagnosis of diabetes.
Well, talk about ATM for a second. We jumped there. Now, ATM, I assume, is not where you don't mean by that you go get some cash.
No. ATM actually stands for ataxia telangiectasia mutation, which you see why it just says ATM, right? So ataxia telangiectasia is another disease, but it's a disease that quite frequently has people who develop diabetes. So from that association, they said people with this disorder almost always have diabetes. So what is it that's going on that makes that association? So what ATM is it's a gene that's involved in the regulation and the effectiveness of metformin. So metformin is a drug that's been around for more than 50 years as an official drug. However, it's a derivative of the French lilac. So this was a drug, this was a nutraceutical or a plant-based therapeutic back to the Middle Ages that helped people regulate their sugar before they realized that they needed to have regulation. So it's a drug that's been used for a long time. It is quite frequently prescribed as the first line of therapy for those with a type 2 diabetes diagnosis. So it's almost always the first thing that we try. The glycemic response to this drug is very different among people in the population. Some people respond really well to it. Some people don't respond to it at all. So the idea here is if we look at ATM, it helps us to understand whether or not a patient would respond to metformin. So that would be valuable if you have someone who's got a variation or mutation in ATM, you would likely not prescribe metformin. What we'd like to do is be able to put someone on therapy that's effective as early as possible. Right. The longer it takes to get someone on the correct therapy, the more potential damage that you could potentially do to the kidneys and the rest of the tissue. So it's a gene, and there's variants around ATM that affect the response. It also affects the ability of to get into the serum. The one that's been most likely or most often associated is one, RS number here is 112.12617. Most of the time we just do the 617. So if you have a variation in that particular SNP, and what a SNP is a single nucleotide polymorphism. So if you go back to that diagram that you had before the DNA, if we were to take a gene and one little point, one little letter out of that whole gene is changed from a G to an A or whatever, that's a SNP. That's a single nucleotide change, right? That can have no effect. It can be entirely silent, or it can have a dramatic effect because it changes the way that the protein is folded, and the folding of the protein drives how it functions. So this one has a significant impact on metformin and the ability to actually benefit from metformin. So what metformin does is it's kind of the opposite of what TCF7 does. Metformin manages the production of glucose from the liver. So if you can modify the production of glucose, right? So instead of making more glucose, your levels don't go up, you're able to manage this a little bit better. So ATM has had a lot of investigation. There have been a number of really strong studies that have shown that, if you've got, 1 or 2 copies of this, because we know we have got 2 copies of every one of these genes, if both of them are knocked out, then you're going to have a more dramatic effect on regulation of glucose. Right. If you only have one that's dysregulated or knocked out, then you have somewhere in between, right? So it's another very important marker that if you're diagnosed, newly diagnosed with type 2 diabetes, and the consideration is to put you on metformin, it would be wise to check to see whether or not you have a normal functioning ATM gene.
Absolutely. Fantastic. And then haptoglobin, which you and I know this is one of our favorite subjects. We discuss it often.
We've spent a fair amount of time working on haptoglobin over the last few years.
Yes, we have.
So
first a lot.
This is an example of, so we've gone through, here's a gene that might lead you towards the development of type 2 diabetes in the TCF7. Then here's a gene that might help understand how to better regulate the disease if you do get diagnosed. And now we're looking at a gene that might have an involvement in aggravation, things that are going to come on top of the diabetes. They're going to further alter your ability to stay healthy. So haptoglobin is a protein that was discovered back in the 20s, and it's an example of a duplication. So haptoglobin in every other organism except for humans has 5 exons. In this particular case, we had a hiccup or duplication in exons 4 and 5. And the thought behind this is, at one point in our past, this duplication conferred an improved ability to fight infection. So for those people who have this hiccup, this duplication, it's given them an extra bit of assistance in trying to clear and handle infections. That could
particularly in infections, if I understand it, probably occurred in South India 10, 12, 15,000 years ago. Yeah. And it particularly helps against the kind of viruses that we're dealing with now with things such as COVID or coronavirus, but it was malaria and other infectious agents that the body wasn't prepared to deal with, so they made it more susceptible to handle those types of infections. Is that a good statement?
That's right. So the idea here is that this was a selective advantage for us in times when infectious disease was a major threat. Infectious disease now, for the most part, this particular pandemic aside, infectious disease is not a major cause of illness or death for the most part, because we have treatments, we have antibiotics, we have ways of supporting people. But and many years ago, when this duplication first occurred, we were caloric restricted. It was tough to find food. So, we didn't have the issues then that we have now, where we have an overabundance of nutrition, and maybe even not nutrition, but we have an overabundance of calories. So what happens here is HP1, their principal job is, in addition to having some protective advantage for infectious disease, is it's intimately involved in the clearance of iron. So our red blood cells have an iron at the center of it, and that's what allows us to carry oxygen and CO2. Over a period of time, those red blood cells just naturally go through their life cycle and they die, for lack of a better term. So the iron that's at the center of that red blood cell has to be recycled. So haptoglobin's job is to attach itself to that iron, carry it to the liver, where it can then be produced into another fresh new red blood cell. What happens, though, is if you look at the diagram there, HP1 essentially has kind of a barbell shape. And that barbell shape allows it to squeeze its way into tissue. It allows it to find the iron that's circulating free in the bloodstream. The HP2 and HP21, because they have all these branches on them, it doesn't allow it to move into the tissue as easily. So it has a more difficult time finding that iron. And what we've discovered is that iron has a tremendous ability to cause reactive oxygen damage. So it leads to all these free radicals. So you've undoubtedly seen, a lot of times we get all these cosmetics and you should do this and you should do that to avoid free radicals, right? Free radicals are a bad thing when it comes to your vasculature. Well, iron is one of the driving forces for production of free radicals. So what it can do is that iron can oxidize your LDL, your bad type of fat. And when it does that, it causes an inflammatory reaction. So the macrophages try to go and eat up that oxidized lipid. And when it does that, it starts producing all kinds of chemicals. Those chemicals further aggravate your vasculature. They cause endothelial dysfunction. They cause heart attacks in people with diabetes. They cause stroke in folks with diabetes. They make your HDL, your good lipids, they make that function worse. So it's been discovered over the last 20 years or so since this marker has been brought back to the surface, that looking at patients who have type 2 diabetes, especially those that have an A1C above 6.5, have the highest risk for a complication from diabetes, like a heart attack or stroke. The good thing about this is once you've determined what your phenotype is, once you've determined which bucket of haptoglobin you're in, whether you're HP11, HP21, or HP22, we know what we can do about that to help people get back to a healthy vasculature and a healthy clearance of iron. Right. And as it happens, if you're a HP22 and you have type 2 diabetes, you're 5 to 10 times more likely to have a heart attack or a stroke than someone who is an HP11. So that's a significant difference, right? And we know that folks with diabetes already have a heightened risk for heart disease. So this elevates that even higher. So it's really important to look and see if you can determine your haptoglobin genotype, phenotype, and then implement the therapy that you need to reduce that risk. And as it happens, the therapy is very simple and inexpensive.
Which is?
You have to implement a surrogate antioxidant. So that could be vitamin E, which is the best, or vitamin C, which works pretty well, but not quite as good as vitamin E. So, but it's important that you know which bucket you're in. And the reason for that is if you are 1-1 and you take vitamin E, you could in fact push yourself towards the very thing you're trying to avoid. You can cause yourself damage and result in a heart attack if you take the vitamin E when you're not.
In large doses.
In large doses, right. So, I mean, what you get from your diet, avocados, that sort of thing, healthy diet, is not going to result is not going to result in a problem. But if you were to go take a handful of vitamin E, and you get 600, 800, 1,000 units, that's conferring some additional risk to you because you don't need the extra help. Your body is perfectly capable of recycling the red blood cells.
Right. But the 2-2s, which, by the way, full disclosure, I'm a 2-2. I come from a family of 2-2. So everyone on the channel knows my Aunt Martha listens, and we love Aunt Martha. Aunt Martha, you're probably a 2-2 as well. So number one, try to avoid diabetes. 2, keep your blood sugar A1C levels below 6.5. And number 3, get a bottle of good vitamin E and take it and cook with things that are, for instance, I think palm oil is one of the favorable oils to cook with that, really gives you favorable vitamin E. So it's probably a good thing. If you don't know your answer, still cook with palm oil, eat some avocados. At those levels, it doesn't do the therapeutic solution, but it certainly helps move you along that path. And at those levels wouldn't hurt you if you're a 1-1 or a 2-1. It wouldn't hurt you at all if you're a 2-1 because they have an increased risk, as you see as well with haptoglobin. So that's very interesting, Dave. We covered these here on the treatments. Those we'll later post this presentation for those that want to look at it and download it. So talk a little bit about then these put that the reason you chose the three to talk about the three is combining those gives you that full picture, correct? Those three genes that we're discussing.
It tells you if you're trying to be proactive about your health, say you're healthy today, but 5 years from now, who knows? If you were to do all three of these tests, you would know, do I have a heightened risk of developing diabetes? Yes or no, because of TCF7L2. Should I develop type 2 diabetes? Will I respond to will I respond to metformin?
Right.
Meaning that my ATM is normal and my ATM is dysregulated or not functioning properly.
Right.
And then further, okay, if I do develop it and I respond to metformin, at some point in the future, you start to have oxidative damage. It's inevitable. So is that oxidative damage as a result, or at least driven in part by a change in my haptoglobin level?
Right.
And haptoglobin has other features. It's involved in other things. So it's involved in stroke. It's involved in some kinds of cancers. It's involved in celiac disease, perhaps, right. Because the precursor for HP2 is a protein called zonulin, and what zonulin does is it causes ordinarily the epithelium in your gut is very tight, so the cells are right up against each other. They don't allow any material to cross over. With zonulin, those gaps open up, and it allows material from your gut to go in across your gut lining and into your bloodstream.
Correct.
So that means that you could respond to wheats, you could respond to all kinds of allergens that you intake as part of your diet, but they aggravate you because they can cross from the gut and into the bloodstream. So there are a number of other reasons to look at haptoglobin phenotype, genotype, other than diabetes. But if you have a family risk, or if you think that you just want to be kind of ahead of the curve and make sure that you're proactive in every way you can, diabetes has a tremendous impact on us, right? Both personally, but then also as a society and as a medical issue. Diabetes is a big issue for us right now.
It may be the biggest, and probably associated more with the deaths from COVID than any other risk factor, probably.
It has been very impactful in that, yeah. So we have seen quite a lot of people with cardiovascular and diabetes and the like have trouble responding to COVID, and part of it's because the cytokine storm that gets generated during a COVID infection. Well, as it happens, haptoglobin is turned on and turned off by inflammation. So the more inflammation you have, the more haptoglobin you try to produce. Right. If you don't produce the correct kind, in the sense that you don't have the normal, fully functional haptoglobin, you just drive more and more inflammation.
Right.
So it turns out to be a biofeedback loop that never turns itself off.
Correct. By the way, on the zonulin and the tight junction gut impact, I've seen a few weeks coming. There we'll do a YouTube live show. One of our shows with Dr. Brewer will be on that. I've read through his PowerPoint presentation. I'll share that with you as well. Be interesting to get your feedback. But just as a quick advertisement, that will be coming soon. Well, fantastic, Dave. Thank you very much. That's great. Let's jump to the Q&A with our readers and some discussion. So, Gilbert, if you'd be so kind, if you can knock out the my share, or I should stop it, I guess. And we'll go back to reading these questions. So we'll jump up and start reading questions this morning. Gilbert, if you can keep up with me, we'll start. Dr. Brewer said good morning. That was me typing that in, doing a test. My Aunt Martha says good morning. Amir Al-Gayar says hi from Germany. And Vagabond Sojourner says, what do you think about exercising while in a fasted state? Of course, Dr. Brewer's not here, and I know that was intended for him. I do fast. I fast fairly regularly, 80% of the time. And when I do exercise, I do prefer to exercise. Well, let me rephrase that, Dave. I don't prefer to exercise ever. But when I exercise in a fasted state, although your strength, you can feel the diminishment of that exercise. I get better results, actually, from that. I
do most of the time when I'm cycling or doing things, it's in a fasted state.
Yeah, by the way, Dave's one of those super cyclers. I was actually at home a couple of years ago, and a guy rode by and looked at me, and I looked at him. It was David riding a bike. He was a long way away from home riding his bike. So Dave's a super cycler. Bart Robinson says, greetings, everyone. Vagabond Sojourner asked the question, how important are the LDL particle numbers and associated with HDL trig ratios and insulin resistance, particularly SDL, LDL number? I'm not going to answer that question. That's specifically for Dr. Brewer. Clinically, we're real careful, although Dr. Vigors could answer that one. I'm gonna skip that one, but go to your second question, Vagabond. Come back and ask that question later for Dr. Brewer. But very interesting, your next question, Dave and I do, particularly Dr. Vigorous, has real insight into that. Have you heard of the Boston Heart Lab inflammation test called OXPL on the ApoE particle? It's oxidized phospholipids on ApoB. Thoughts? We do have thoughts on Dave. We have heard of it. In fact, you've been very integrated in that. So give me a quick 2-minute. I know we got a lot to cover in 20 minutes, but give me your quick summary because it ties very much into the same mechanisms as haptoglobin.
It does. So I've been working with a group that's been in partnership with the gentleman, Dr. Sam Samikas at UCSD in California, who developed this assay. And this assay is likely going to be the most powerful cardiovascular assay in the future here. As we start to get more and more exposure to this assay, it's going to be more powerful than some of the other tests that we do for cardiovascular disease, like lipids and triglycerides and that sort of thing. This one has much more impact because oxidized phospholipids lead to a significant secretion of cytokines and inflammation. So you really do need to know if you have oxidation on your lipids, because that drives through macrophages, it drives your other immune cells to try to respond, because it's essentially a danger signal. It's a damage signal that you're responding to. So,
isn't it the same mechanism when your cells lyse, and say I'm a 2-2, and my cells lyse, that free heme now is running through my bloodstream. Right. And it has an affinity to attach. So if the haptoglobin normally with that barbell would go in and attach to that heme and clear it, carry it back to my liver and recycle it. But it looks like a crab. I think you've described it before, that 4 or 5 armed creature called haptoglobin 2-2. There's no binding point for the heme. And so that heme has an affinity for the HDL. And it does the same thing we're looking at in that OXPL. It starts the oxidation process of an HDL. It actually would, I guess, convert it to a LDL-like particle. The same as looking at your ApoB oxidation. There's no good oxidation in the bloodstream, I guess. So
not really, no. You need to have mechanisms to eliminate it, right?
Right.
And if you don't have a good solid mechanism to eliminate, then you're going to get tissue damage, vascular damage.
Right.
This gets into the blood wall or the blood vessel wall and causes this growing inflammation. At some point or another, that inflammation ruptures, or that plaque ruptures.
Right.
And as soon as that plaque ruptures, your body tries to stop, that injury, and we make a clot, and that clot then travels to the heart or travels to the brain. So I'm confident that OXPL is going to be the next major marker for cardiovascular disease.
So Boston Heart, good lab out of Boston, hence the name. We're rocket scientists here, but Boston Heart does a venous pull, which is a full tube of blood. I think there's a couple of other labs I know that are exploring microvascular or micro testing, using a finger stick, or maybe even, could you get saliva? Could you use saliva theoretically? I know you're an expert on that, but probably
more of a challenge to use saliva. But, for some people who are not close to a draw center, not close to a patient service center, or who are just, not happy about the prospect of getting, a needle in the arm,
right.
This can be done with a small volume draw, either dry blood spot card or one of these little microcapillary tubes that you can draw, 200 microliters of blood, send that off, and you get a determination.
Right.
So, I think this is going to be a powerful tool moving forward to help us better understand who's at highest risk for CVD.
Right. All right. So we'll keep moving. Question. Robert Weiss said, good morning from North Georgia mountains. Good morning, Robert. Then O Brothers, thanks guys for shedding a light on health. We need to shine a light on all sources of inflammation, which gives names to like diabetes and heart disease. And by the way, cancer. Cancer, the mechanism of most of the cancers is inflammatory based as well. It's rheumatoid arthritis. I mean, practically almost all our diseases are some form of inflammation. Would that be a true statement?
What I didn't mention with ATM is also involved in response to certain types of cancer.
Right.
Because of the mechanism. So ATM is a gene that regulates another switch. And in this case, it's a phosphate. So phosphates are quite often on and off switch. You add a phosphate, it turns things on. You remove a phosphate, it turns things off. So that's what ATM does. And that is also important in your cell cycle. So as your cells go through their replication, they go through their normal metabolism, they have to turn on and turn on things. So ATM is intimately involved in that as well. You come into these connections continuously, these connections across different disease processes that are related to inflammation and cancer.
Right, exactly. O Brothers also says it's amazing how people are being misinformed to cause themselves inflammation with diet, etc., further down the road diseases. Absolutely the case. I agree. Buyer beware. What was those cigarette commercial Chesterfields? They satisfy. So marketing over the years, food, other organizations will market to you services, food, activities, behaviors that may not be in your best interest.
Yeah.
Again, one more question from O Brothers, a comment. As part of yearly physical, doctors should mandatorily be testing patients with a full spectrum of tests for inflammation markers at least once a year. Although I would agree with that, I'll be practical, Dave, and get on my soapbox. I love doctors. Doctors are my world. You are one. We're all part of that cycle, but the current structure of how a clinic runs makes it difficult for most of the providers you would see to order that much information and then to know what to do with it and then to do it. And it just creates all kinds of liability exposures. And so that's why you have to find a provider like a Dr. Brewer, Kevin McCord, or Belle Donine, anyone from that functional medicine, those providers. It's not that the other doctors aren't good doctors, just the mechanism. I keep wanting to say mechanism because you said it, but the process of how a clinic runs. Once you're in this business and once you understand it, it becomes impractical unless you change the entire structure of your practice to slow down and do the things that you have to do because it's so complicated. And exposes you. Once you order something as a physician and you get that information in, you have to do something with it. And so if the patient hasn't complained, the tendency is don't go looking, don't go digging around for things that you don't know what to do with when you find it.
You have a provider who studies this and understands what to do about it and has the time to go through and review a patient's results and then counsel them on what's the best thing to do next, right? Traditional medical practices have to see so many patients during the day that they just don't have the time to devote to each individual patient.
Right. And so much of this is not reimbursed. So these tests, you and I have lived through that. We together have developed tests. You're the brains and I was the guy that went out and tried to sell it. But we would, it'd be perfectly usable a test such as ATM, but you may get the payers that say we're not going to reimburse for that. So then the patient gets a big bill. Therefore, the insurance company won't pay. The patient's mad, and it's just a cycle of disruption in the patient's life that most clinics will say, well, we just don't want to chase that. So that's unfortunate. It's the system in which we're in. Kevin McCord says, thank you for navigating this topic today. Dr. McCord's Northern Kentucky provider. He also is a prevention specialist. Appreciate the information. Important to remember how our lifestyle choice influence our epigenetic expression. Absolutely. Pancakes. I love them. I'd eat them every day. I'd eat them 3 times a day. I can't eat pancakes because they will trigger a lot of bad things. So I just put my syrup on biscuits instead. All right, so John Tocho says, wonderful overview. Thank you very much. Thank you, John. Robert Weiss says, this is crazy interesting, multiple play to really understand it. Well, see, that's a combo word. I'm the crazy, Dave is the interesting. Works out great. How would I know how to get tested for HP11 or HP22? I know Lifetime Sciences test for it. I don't guess anyone else has brought that test up. I'll brag a little bit. I know lots of labs have tried to get this test up and marketed. It's a very, very difficult marker to find. And the only one that I know was the one that we developed together. Dave did all the science, did all the work, and it took us a lot of time and a lot of money to figure that out. And since then, I just don't think labs have figured out how to identify it. We have some theories how to maybe do it now, and that may be coming soon. But without giving away your secret sauce, you actually filed the patent around that and so forth. It's a very difficult marker to identify.
Yeah, it'll be coming back soon. And likely a two-step process, one for a screen and then a second for a confirmation. So a quick and easy screen, yes, no, and then a confirmation, yes, in fact, you do have this genetic variant. So
well, when that becomes available, completely anticipate you'll come back and talk about how to do that, when to do that, where to do that, because we'd be greatly interested in that. We've said before, I think everybody ought to be screened for haptoglobin. You know, why would you screen for all these other things we do it's such a significant piece. Dr. Brewer has chimed in and said, hello from vacation at Orange Beach, Gulf Shores, Alabama. You guys are great. Hit the like button. Well, that is great. We do need you to hit the like button. Particularly all you guys need to hit the like button on a day when Dr. Brewer isn't here. So we can say, see, Dr. Brewer, this is how you do it. We can play some of that music in the background. Thank you, Dr. Brewer, for taking time out. I assume he's landed and watching. He deserves the vacation, a little bit of rest. I had a whole list of things. Just to let y'all know, I'm a slave driver. Dr. Brewer, I knew he was flying. I knew where he was going, and there was some work in the area. So I had someone meeting him at the airport to get some paperwork signed, and he went, James, you missed the whole point of this idea of vacation. I said, well, it should be easy. It's just a few things to sign, a few meetings to have. Can you run by a couple of clinics and so forth? But he's enjoying the Gulf Coast. We are going to open a series of clinics along the Gulf Coast. There'll be Medicare Advantage primarily, dealing with the Medicare population, prevention based. We're very excited about what that business model will roll out and look like. And so if you're in that area, Western Panhandle of Florida to the Eastern Shore, Mobile, Alabama area, reach out to us. We'd love to talk to you about being patients in the clinics down there. And don't forget about Patreon, John Tocho says. That's right, Patreon. We need the help. We need the support. We just can't live off of Dr. Brewer's savings forever. So if you could participate with us and help us get this message out to the world, it does do a lot of good. I mean, there are a lot of people. In my personal life, Dave and I both have some personal connection in that both our mothers passed away way too early from inflammatory diseases. And I wish we knew what we know now much earlier in their lives. I think we could have made a difference. So that, just on a personal level, that's what drives a lot of what both of us do, is our own personal experiences of seeing someone we love dearly suffering with these kinds of diseases and not knowing what we know now, and even to an extent help create and drive out a result of the passion that we had for helping people with these diseases learn and get better and well. And so your loved ones will depend on it at some point, and maybe even you one day may benefit from what we're developing in these channels. So Bart already hit like and says, had a great time, Doc, down at the beach. So Bart says, enjoy yourself while you're down there. Dave, one quick question, and then those are all the questions we've run through them. Thank you guys for listening. Thank you for participating. I'm interested about haptoglobin, since we brought that up, too. Because of its impact on things such as malaria and others, might it be that some of haptoglobin may be playing a part in the defense against COVID-19? I keep laughing and say I haven't gotten the vaccine, I've taken ivermectin. I take a lot of vitamin D and do other things preventatively. And I have been around it so much. I've been in it, been around it. Everyone close to me has had it, but I haven't. And I haven't tried not, I didn't purposely ran in and, tried to lick a frog or anything with COVID. But maybe those with 2-2, because we're protected against those types of infectious diseases, maybe it has less of effect. Any thought or research seen in that mechanism?
They haven't gone down the road with research. I've talked to a couple of groups who are thinking about doing that. One in Germany is really interested in the connection between haptoglobin and COVID. And so I think they were looking at other proteins within the lung, but obviously, you do get some coagulation and some blood disorders with COVID-19. So there may very well be some connection there that we can tease out as we start to understand more about all the different things that can happen as a consequence of the infection, because it's so varied. You get some people who have no effect whatsoever, and then some people who get, these coagulation disorders that are fairly significant. So I do think that haptoglobin might play some role there. Whether it's a protective role or a complicating role is still not really that clear. I suspect that it might be a little worse for somebody who's haptoglobin 2-2, but that's just sort of like a gut looking at some of the literature on complications. Right. But I don't know. We'll have to see if someone takes up the staff and tries to look retrospectively back at patients who had a severe outcome and whether or not they were HP1 or HP2. I'd like to do that myself, actually.
Right. It would be a too difficult study to conduct.
No. If you've got, biobank, a lot of these labs, I know one lab that I've worked with more recently here has saved all of the samples that they've run so far, and they've run more than 7 million COVID patients. So it would be interesting to go back and, take 5,000 of those patients and test them for HP2. If you had the clinical data to also associate that they had
severity,
not an easy course, but, if they had a uncomplicated course versus a complicated course, maybe we could tease some information out about that and understand better. But certainly haptoglobin 2 does protect against certain types of viral infections. It protects against certain types of bacterial infections, and it makes it worse for some. So, going back to this diabetes things, oftentimes you have soft tissue infections, right? The bacteria that cause those soft tissue infections actually rely upon iron. So if you're incapable of clearing iron, you're giving more nutrient material to the bacteria.
Yeah, you're fueling.
Yeah. So, those patients who are HP2 that have, soft tissue injury or soft tissue infection, they may be, leaving enough iron in the tissues to support the further growth of those bacteria. So, there's always in everything, there's a seesaw balance.
Right.
And, if you know what your genotypes are for these different markers, I think you can do a little bit better job of being proactive and heading things off complication-wise.
Right. Absolutely. Well, Dave, our time is up. Thank you very much. Great show today because of you, and we appreciate you being on. We look forward to having you back again soon. We encourage our audience. Next week, Dr. Brewer will be back, and over the next several weeks, we have a lot of great topics that will be coming. So share, and we look forward to seeing you all again on this channel. Thank you, Dave, and everyone have a great day.
Thank you. Appreciate it.